Hello everyone. Today's video is related to drug design, development and discovery process. We all know that it is a tedious process and involves a lot of resources. So it becomes our responsibility to study each and every aspect in detail, thus making it a lot more complex. In order to design any molecule, any drug molecule, we need to identify which particular disease we are targeting. When we try to see the disease mechanism, we come across a number of molecules which are not functioning properly, thereby leading to some sort of abnormal activity. It could be enhanced functioning or disrupted functioning. Once we are able to identify the target molecule, we then try to figure out which lead molecule can bind to the target molecule, and thereby that complex will be able to show some sort of stability or differences, leading to the kind of response which are required for a normal functioning. If everything is going perfectly fine, we tend to move on to animal models, and then if the results are positive, we even go for clinical trials after getting due permissions from the different regulatory authorities. From this slide, we can see that identification of the lead molecule is again not a simple task. It involves studying of thousands of molecules as is visible in the slide. During the drug discovery and screening process, we usually approach through high throughput screening, and the whole sole purpose is to identify the right molecule after target validation. We further tend to optimize the lead molecule according to the requirement using combinatorial chemistry approach, structure-based drug designing, and then seek whether the target molecule and the lead molecule are showing the right kind of ADMET properties or not. We all know that absorption, distribution, metabolism, and excretion, along with toxicity studies, are basically required in order to give the first level permission to a drug molecule to proceed. If we are getting positive result, as we can see, we can move to step 4, which includes clinical trials. Once we are getting positive results in the clinical trials, after approval from the authorities, the company will be allowed to introduce the drug to the market. Now, all this process can be further divided into 3 main divisions, like the research part, the risk assessment part, and how do we actually manage the risk. So when we talk about research, it is not just confined to the laboratory approaches. It includes how you are able to merge the different data set, how you are able to understand what kind of differences are happening, what is actually happening when the lead molecule is binding to the target molecule inside the cell. And obviously, we need to take care of the genetic approaches also. We need to see how the gene environment is responding to the kind of interaction which we are looking for. And in the process, we need to assess how toxic the drug is, to what level of toxicity we are able to allow or permit during this particular stage. We need to see what